freaked me out.
I know, I thought that we had homework today. I was like that.
So tonight we begin 2 nights of my favorite topic, which is special senses. So first off, we have to make a list of different kinds of receptors that we're going to talk about tonight and Monday. So if you remember, way back in 231, receptors and sensors are basically interchangeable for this class. So let's go through the different types of what they are. So in lab, you learned photoreceptor or photosensor. Can someone tell me what that detects? Light. That's a light detector machine or anything. Okay. So we have photodetectors that detect light. What part of your body would I be talking about when I do photoreceptors? Eye works, retina works, fine. So another one. In lab, you talked about mechanoreceptors. What did mechanodetector receive? Pressure or vibration. So we can do pressure as in touch, or vibration. Something mechanical, touchy-feely. What part of your body would I be talking about if I want to talk about mechanoreceptors?
Your ears.
My ears, my skin, those kind of places where I have touch and vibration, which we'll probably do on Monday. Then our next one should be self-obvious. Chemo. What am I detecting when I have a chemoreceptor?
Chemicals.
Chemicals, just like the name says. What part of your body would I be dealing with chemos?
Your skin. No, organs.
Mouth, nose, places where I smell and taste different chemicals in the food. So these are the big 3 that we're going to talk about this week and next. All right? So we're going to start with the photoreceptors, and then next week or tonight maybe that one. So we're going to start with the light receptors. So thou shalt know, PCC says, the route of light to your eye. So since that's in lab tonight anyway, let's put it on the screen. There's the picture, and you're supposed to pretend you know the pathway. We're going to write out what the light would go through between Mr. Sun and the photoreceptors. So what's the first thing the light would hit? Cornea. Cornea is a good answer. So here we go. Cornea. We'll talk about in a minute. What comes after the cornea? The answer's on the screen. Okay, the cornea is here.
Aqueous
here.
humor.
Aqueous humor. That's a fluid. Remember the humors from way back in the Dark Ages? They're here. So literally watery fluid. Okay, it's a fluid. What am I going to hit next after my aqueous or aqueous humor?
Your pupil.
My pupil, which is the hole. You actually don't go through. You go through the hole. The iris would be the thing. Okay? Keep going. What comes after my pupil?
Lens.
The lens. Okay, we'll go this way, that's okay. What comes after a lens? What is here? Vitreous humor. Glassy goop. Literally. Your brain put a U in it. Okay? Then what? What comes after my vitreous humor?
Your retina.
The retina. Okay, we're going to stop there for a second, because the retina is where the photoreceptors are. So the photoreceptors are in the retina. The retina is the place, the photoreceptors are the things. Do any of you remember what a 35mm camera actually was like when you had one? Yeah. The film? Mm-hmm. Do you buy film still? Do they exist?
Yeah.
Remember the film was in the back of the camera, right? So the film, the photoreceptors in the back of the eye, the image, the lenses are in the front of the eye, just like an SLR camera. It's the same logic here. But we're not done yet. Because we got to the receptors, but where do you actually see? It's not there. In your occipital lobe. So we got to get to the brain. So we got to our photoreceptors, and we got to get to a brain. So we use a different color to head to my brain. Red. How do I get from my retina to my brain? What's next? Look on the screen.
Optic nerve.
Optic nerve. Remind me from your quiz coming up, what number of cranial nerve is that one?
2.
Number 2, right? olfactory optic. That's a sensory nerve for my vision. So the optic nerve is then going to send it to my brain. And what part of my brain sees?
Occipital.
The occipital lobe of my brain. So the reason I picked two different colors is something Saladin has a hard time explaining. But there's two parts to all these. One is to get to the receptor, and one is then to get to the brain to interpret what the receptor's saying. So depending on your philosophical school, you can say your sight is actually in your brain. The green is just the camera. The red is actually where you see. It's like your little camera. You plug it into the computer to download the pictures. So I get to my eye, I have an image. The brain then has to interpret the image and make it all what I can actually understand. Make sense? Yep. So there's the seeing and the interpreting part. Make sense? So what we're going to do is we're going to focus on the photoreceptors first, because that's where we're going to see. So before we do that, we got to figure out this other stuff. Why have a cornea and fluid before you get to the photoreceptors? Why not just have an eye spot like a worm?
For protection.
Refraction. What's refraction mean in English?
Bending of light.
Bending of light. In Biology 112, what did you use in lab that had a bunch of knobs?
Microscope.
A microscope. And remember your microscope had lenses, and when you got really high powered, you had to add that drop of stuff you forgot to wipe off and got in trouble. What was that stuff? Oil. You had to add fluid
Immersion oil.
between the lenses, right, to help it refract. So these three things here, the cornea and the fluids, actually four things.
Ooh, poor thing.
That's all for focusing. That's to help you focus the image on the camera in the back. All right, so you have a focusing mechanism, fluid and lenses. You have the photo image, and you have the brain. Of course, they're good? Mm-hmm. So what we're going to do is I'm going to walk you through some animations of this process. So I'm going to show you how I got here. Hold on. So what you want to do, by the way, I don't work for this guy. But the best website in town, if you're bored, I love this guy's name, Tutus Villas. That's really his name. You can't beat that, Tutus Villas. But he has more there than you can imagine on seeing, hearing, way beyond what I need to teach you. But if you go to Tutus Villas and type in something, here's his website. He's in Canada, of course, he's a Canadian. So here's his Canadian website. If you scroll down, he's got all these animations and tutorials on everything about everything. Very good stuff. So if you're interested in this, go visit. If you're not interested, stay away. I'm going to use his stuff to teach you some things. So I'm going to go to the eye. Here's my eye. Okay, so here's again the concept of what your eye is doing. So I have a camera. I have focusing stuff in the front. I have the film in the back. And I have the computer to interpret it downstream. Right? So what we're going to do is do that. Here's my lens. So let's figure out why I have two lenses. I have a cornea lens and a lens lens. Why do I need two? By the way, there are two knobs on your microscope for a reason.
To bend the light.
Focus and...
Coarse and fine. If you look here, the cornea is basically your coarse focus. It's fixed. It doesn't move much. This one you can adjust. That's going to be your fine focus. Just like a microscope. But microscopes are copied from your eye. In the sense that you need two lenses to make the image clear. Right? Unless you're an eagle, they're different. But here's what your lens is doing, trying to fine-tune every time you shift your head. You have to bend the lens, shape it, to make it focus. The cornea doesn't move, it sits out there. The fine focus has to change. Right?
The fine focus
was just formally the lens. So the cornea is rough. So, another view of it is this. I don't know if you can see that, okay. So you're bending the lens, to make the image stay on the photoreceptors. Right? So I love this little thing. Here's the picture of the taxi. And here's what your lens is trying to do, is to keep that taxi in focus on your eyeball. Right? So if you're like me and your eyes are getting worn out from college, you then have to add an artificial lens in front to bend the lens before it bends the lens, right? So as you get older, the lens is harder to move, harder to work. An old microscope is Christie, so it's harder to fine-tune your image on the film, right? Just like a microscope would be. So here's here I am, I can't read anything. So I can give you a fakey lens. That's worse, that's better. And it's going to help my lens do its job. Make sense?
Mm-hmm.
All right. So why do you need an iris then? What was the iris for, the pupil thing in your microscope?
Allowing light in.
The light control, right? Same thing here. Is the iris's job is to keep the light from being too bright, too dim. So again, a microscope and your eye are the same. Where it gets different is this part. These are the photoreceptors in the back of the eye, in the retina. So what I'm going to do is I'm going to shift gears here for just a second and show you a static image of that first, and then I'll go through the animation here. Here is your retina. So to get your bearings, the light's over here. Zooming in through all this wiring, and it's hitting the retina, which is in the back of the eye. And then notice it says receptor cells. These are the photoreceptors, or the sensors for what I'm seeing. Notice there are two kinds there.
Rods and
See them?
cones.
Rods and cones. So why do you need two kinds of photoreceptors?
For color and for black.
Color and black and white. Very good. So we're going to do a little activity on the rods and cones first. It isn't in your packet, because I just wrote it today. And we'll see if it is any good.
What did you call the thing on the bottom of the microscope?
Iris or diaphragm. Or you can be fancy and say iris diaphragm. Or the condenser adjustment knob, which is what they say in your 112 letter. Rod cone thingy.
Are cones color? Do you remember?
I don't remember. I'm just gonna wait for my thoughts.
So what you're going to do with your phone and your friends,
Looks like we're not. Looks like we're gonna have to do it ourselves.
top, this paper I just gave you, that asks the differences between the rod and the cone. I don't care about how they look, right? That's how they're made. But see if you can figure out which one does what in terms of, is it color or black and white, response, sensitivity, acuity, convergence, and all that. And then I'll give you about a minute or so, then we'll go over it and make sure you're okay. See if you can figure out the difference. And I'll write it up here.
Okay, rods handle vision in low light. So rods is low light.
One more of the other.
Well,
Cones.
Okay.
Forgot.
Cones handle color vision and detail. Okay, so cones are color.
okay, so rods are black and white.
I would guess that the.
They do not mediate color.
Acuity? Acuity.
I would guess.
I don't know.
Is that detail?
I believe so.
But they all detect light. But one of them detects color.
Cones are color, rods are black and white.
And
Sharpness. Yeah, you're right. Okay, so
that's
cones.
And then
Yeah.
there's cones.
So high. Cones is high.
No, wait. There's more rods.
Okay.
Color blue, chapter 17, page number black 558.
low light. So they,
Okay, so
5:15.
rods.
558.
5:15, okay.
Color vision is in
Cool.
Lots of black and white.
Function specialization of the rod and cone system.
Color and?
They are highly sensitive to light, the rods are.
So response time fast.
Yeah.
no?
Cone slow.
Yes.
Yeah, this is.
So the cone system has a very high spatial resolution, but is relatively is anything but spatial?
Just not linear.
Oh,
You buy it
It's
out?
acuity and convergence.
oh right, okay.
Acuity high.
Oh, I remember. Rods.
Oh, I did do a response time.
They're not the same thing, but same.
Rods are sensitive to light, cones are not.
Rods, yes, rods are sensitive to light.
Yeah, there's okay, don't get too bogged down.
Cones are only good with high light in the environment. Only good in high light.
I did highly sensitive to light. I guess it could be both, maybe.
Since cones are bigger,
Our accuracy is the same.
rods are smaller.
Okay, so
Cones require more intense light, so their intensity is low.
I'm confused between the response time and time.
I haven't really read that yet.
convergence is high in rods and low in cones. Yeah. It says a factor that contributes greatly to their distinct properties.
What's convergence?
A factor that contributes to, wait, okay, it means that they are narrowing in on it or not. Detecting light.
Okay.
Convergence is what makes rods better at detecting light, is what it says.
Gotcha.
So the rods have a high convergence.
Yes.
It shows it just doesn't clear.
Cones low. See, they're both, they both have.
It says the cone system is much less convergent.
Okay, so.
Relatively low. Location on the retina.
Cones are in the center.
Cones are in that dot phobia.
Yeah, so like they're cones and surrounded by the rods.
What's the dot in the eyeball called?
The macula matea.
And on the back of the
Macula matea.
Macula matea and the fovea centralis.
Yeah,
Both
fovea centralis.
Fovea is probably what you mean. It's the dot in the dot.
Dot in the dot.
Or the dot in the hole, yeah.
Away from the periphery towards the center of the retina, the density of rods gradually increases.
It says the response of an individual cone does not saturate at high levels as the rod does.
See how you did getting nowhere. All right.
Yeah.
Rods and cones. So, rods, first row. Color or black and white for rods?
Black and
It
white.
tend to be black and white. Or shading, or, that kind of thing. A black and white movie is your rods, that kind of idea. You can see tones of gray, black and white. Therefore, cones see color. C-O-C-O. Cone color. They're the ones that detect colors in what you see. Remember in the Dark Ages you had black and white film and color film? Your eye came up with it first. You have black and white receptors, you have colored receptors. Okay, response time, meaning how quickly they can detect what's going on.
Rods
tend to be,
faster.
because they can see a lower light, because they're more sensitive.
but they're actually slower to respond.
Really?
Oh.
And you can prove that. I'm going to turn off the lights. You tell me how quickly you can tell what's going on. Right? Because if you go down to the next one, sensitivity to light, which ones are the most sensitive of the rods? Therefore, they're used in dim light. They're sensitive. Dark situations. Cones need bright light. Therefore, they're less sensitive.
So, I'm still not.
So if you turned off the lights and waited 20 minutes, you'd have very good night vision, but it takes you 20 minutes to do that. I'm going to prove that.
I can't see anything.
Yeah, I know. So if you notice, you're having a harder time reading the blue because your rods take a while to get active and turn on, so they are slower to respond.
To
But
darker
when they
colors.
do respond, you will be very sensitive to what you see at night.
Okay.
But there's a difference. They take a while to warm up.
Is it because our eyes are more activated towards color?
Yes. So when I turn these on, you have no problem telling that's blue immediately. The cones can immediately turn on, but you have to have the light to do it. So they're less sensitive in terms of the type of light as long as it's bright. That makes sense?
Yeah.
So in English, at night in dark, you're using rods. In bright light, you're using cones as a general theme. You can use rods anytime, but when it gets dark, it's only the rods. Makes sense? Yeah.
So in elderly people, when they start to not be able to see as well at night, is it because their rods are diminishing?
Yes, mainly because they don't have their
First.
vitamin A. Yes.
Okay.
So let's do acuity. Acuity means how good you are at detecting things. Which one do you think is the best at seeing details? What's
Cones.
asking?
Cones.
Cones, high acuity. Cones are color, they're fast, they're very good in bright light, and they're very detailed. Rods, not so good. Low acuity. Doesn't mean you don't have any acuity, just not as good as cones. So again, if I turn on the light, you can see every little place I skipped with my pen. If I turn off the lights, you start noticing you have problems noticing differences if I turned off the overhead projector. So you can still see, but not as detailed with a rod. Make sense? All right. Convergence. This is a hard concept to sort of to understand unless you look at the picture. Convergence means how many photoreceptors individually get to your brain. So they're converging. So look at the rods. Here I have 3 rods, right here, here, here. If you follow the wiring, 3 rods end up going in 1 wire to your brain. So it's 3 to 1. It takes 3 rods to get a signal to my brain. Does that make sense?
Mm-hmm.
How about over here? How many rods to my brain?
3.
3. It doesn't have to be 3, it just is in this one. So 3 rods combine to make 1 signal to my brain. Look at the cone. How many cones get to my brain?
1 and 1.
1. So it has a lower convergence. It takes fewer cells to get to my brain. So cones have a low convergence. Hence, if you think about it, that's why it has high acuity. Each individual cone is sending a message to my brain, rather than combining or averaging 3 of them to my brain. So low convergence causes the high acuity. It's 1 to 1. The rods have low acuity because they have high convergence. You're averaging more and more things to make a rod go. So you're getting less and less detail. Make sense? It's like on N grade. Each of your grades is high acuity because I'm doing each score individually. If I combine the whole class that y'all get a C, that's high convergence, low acuity. Right? I'm combining everything into one bad score. Make sense?
Mm-hmm.
Okay, so let's do location on the retina. This actually shows, even though it doesn't show it anatomically, where are cones on your retina?
In the center.
In the center, in the center, in the center. All right. That's where the camera reads it. All right, so cones are located in the center of the retina. So when you look directly at something, you're using only your cones. Where are rods in my retina? Outside, periphery is this fancy speak. Outside, exterior, radius, whatever you want to use. So rods tend to be on the outside of your view, cones tend to be in the middle of your view. Right? So, if you're reading a book or your notes, if you're doing it under highlight, you'll be using your color vision. It's very quick to focus. You will have high acuity. You can see every little mark on the paper as long as you look right at it. Then you're going to go home and try this experiment. You're going to turn your eyes and use your peripheral vision to read. And when you get a migraine in 5 minutes, you'll realize because of this, is that the letters are blurring because you're taking all the rods from the outside of your view and combining them into an average sort of picture, which makes it much harder to pick out stuff. Plus, it won't be in color. Try this at home. This is a beer dollar test. Take something like this and hold it out there and tell me what color it is. It's black because I can't see the color. It's only green when I put it about here. Because I have to be in my center view to see the color. Periphery does not see color. I won $1. All right. Never bet a biologist. All right. Make sense? All right. You can try it at home. Yeah. Just keep moving out your peripheral vision and realize it goes from green to whatever. It goes black. Well, let's do time to recover, the last one. Which one, you know this from a movie theater. You go to a movie theater, they turn off the lights. You're going to turn off your cones and eventually turn on your rods. Then you turn the lights back on. How quickly can you see color again? It hurts, but almost immediately, right? These recover quickly. Cones recover quickly. Otherwise, every time you blinked, you'd be blind, which makes no sense. Rods, though, recover relatively slowly. It takes them longer to get up to speed. So if I turn on the lights and then turn them off again, you'll find that you can't see for a few minutes while the rods readjust. So these tend to be slow recovery. Recovery. These are relative terms, right? It's still fast, but. So in a nutshell, cones color, bright light, high acuity, quick center, and rods are the opposite. Black and white, less light on the periphery, low acuity, slow to deal with things. Go to the theater and turn on and off the lights. You'll get this gist. Make sense? So let's then explore that option. If you understand rods and cones, let me show you an animation of rods and cones. And then I'll give you some animations that'll make you wonder if you're really alive. So, here's the retina. So here we have some cones with their pointy hats. That's cool. Here's a rod, you have a square hat. Okay, there's these other cells. Okay, that kind of combine everything. So you're taking your photoreceptors and you're converging them, merging them to get to your brain. And the number of mergers sort of hints at the acuity. This doesn't show that, but that's the convergence idea. How many photoreceptors end up in my brain? Okay. So let's explore that color thing. So go here. So here it kind of walks you through why you have color. But here's a picture. It's not anatomically correct. This is your retina, and here are the cones. Hey, why are they only in the center?
Because that's where they're
That's where
located.
they're at, right? That's the logic. So your cones are located primarily only in the center. And if you notice, they have different colors on the cones. Why is he color some blue and some green and some red? Why doesn't he color them all the same?
Because they see different colors.
They detect distinct colors. So I'm going to show you something here to make you somewhat bothered. Somewhat bothered. I stand here. But here are the only three colors your eye can actually detect. You have a blue cone that detects blue. You have a green cone that detects green. Actually, these are wavelengths. And red. That's it. So if you're looking at a yellow thing, you didn't detect it. It doesn't exist. You have no yellow cones. You cannot detect yellow. So how do you know it's yellow?
Because your mom tells you it's
Your mom
yellow.
told you, yes. But how does she know it's yellow?
Because her mom told her.
Where did the yellow come from if you can't see it?
The sun.
Where do you actually see?
Between red and green.
No. Who
Oh.
sees?
Occipital.
Your occipital lobe. So the concept you want to get in your head is that the brain makes decisions about what it thinks you're seeing. So when you're looking at yellow something, whatever you believe yellow is, you're actually not seeing yellow. You're seeing a combination of red cones and green cones turning on. Your brain then interprets that as that must be a yellow. But you don't see yellow. You see red, green, and blue. The brain creates yellow out of the difference between them.
That's why you have arguments about what color that really is.
That's right, and I'm always wrong. I know that. We've been married a long time. I know I'm wrong. Default. I'm wrong. Sorry, honey. Wake up and just say it. All right. Yes?
Why are the primary colors red, blue, and yellow?
Well, that's for paint. In your eye, they're different.
So they, I mean, but why didn't they make them red, blue, and green?
That's color theory.
Home
It's
Depot,
totally
no, that's
different.
paint. Right. But it is different than the paint stuff. But the concept is the same. You can take those same three colors in Home Depot and create other colors by combining them. Your brain does the same thing. It takes how much red, green, and blue in your eyeball is getting sent, and then creates a color from it. So you want to freak yourself out, you never see anything that's yellow. The brain creates yellow from what it thinks yellow should be. So your yellow probably is not anyone else's yellow. In fact, yellow doesn't exist.
Wait, is this yellow?
Is it?
So the sun is ultraviolet, but you see
Do you?
yellow.
You can stay up nights worrying about this, right? You see a yellow sun, but the brain invents that the sun is yellow. It may be green, you don't know. The brain thinks it's yellow. What you see is what the brain interprets from your eye. You don't know what's really there.
That's great.
Yes,
So
you'll
people
stand
that are-
next to this, yeah.
So people that are colorblind, are
Yes.
certain cones not working? certain color cones?
Indeed. Let's go back to this picture. I'll lead you there. So in your eye, you have a mixture of that red, green, and blue photoreceptors. They see only one of the colors. The brain then is interpreting this mixture of who's on and who's off to come up with the other colors. The, whatever. How many colors are there in the world? Who knows? Right?
Zillion.
Right? So colorblind, for example, if you're colorblind, one of those cones isn't working. So the brain is missing that piece of information to determine what color it is. So it goes with the other colors it has. So for example, if you're missing your red cone, the brain never gets the red information. So it assumes there never is a red. So everything's a blue or a green because red doesn't exist.
I have a question.
Yes.
So, do you have certain number of like red cones to blue cones?
Yeah, red's the most common, blue's the
Okay.
most rare. And blue cones don't exist actually in the very center. So you can't see blue if you actually look directly at something.
Oh.
Where the but your eyes always twitch, so you always see it. But, yeah.
The two colorblind, not colorblind circles that you just showed us.
Uh-huh.
If somebody that was colorblind
Yeah.
looked at that, would they see a difference between those two slides?
I have no idea because I'm not colorblind. I don't know. But in theory, if that's what they do multiple tests. The doctor can determine from which ones you see and don't see which cone is not registering to the brain.
So does the doctor have to be able to see all of the colors?
Well, I don't know. That's the question. You know, I look at that, I know what I see. You may be seeing totally different colors because my brain may interpret it differently. But we all learn. That's the question. How do you know yellow's yellow?
They
Because
gave
your
us
mom
a
said it was?
colorblind test at the hospital before
So
we.
you can stay up nights worrying about this, but the trick is you have three colors that you can see. Your brain interprets that mixture as what it is you're seeing. Right? So the cones are relaying information. The brain interprets the information as, you know, I'm wearing teal, whatever it is. Right? Make sense? So here's what your brain does. Your brain takes the three colors and kind of breaks them down into how strong they are. So here's the greens, here are the reds, here's the blues. Remember that yellow doesn't really exist. And then your brain tries to say how far away from each other they are. They call that saturation. No, you never have to know this. And then your brain determines how bright they are compared to each other, and you get 2 million different colors out of 3.
Wow.
That's only in the brain. Your eye only saw 3. Your brain came up with 2 million out of it by interpreting the differences and the shading and the nuances. That's why you go to Home Depot and have to fight about the bathroom.
Seriously. How many shades of gray?
Yeah, the wrong white in every apartment, right?
Uh-huh.
That's the wrong white, right? But that's what the take-home message is: not to, don't ever reproduce that. I'm not asking you that. But the trick is to recognize the brain is interpreting the camera and then assigning a value based upon the information you get. You don't see yellow, the brain sees yellow. The brain invents what it thinks yellow is.
So, can over time, when you get older, can you lose more cones?
Yes, you can.
And
So you can have eye damage and you can start losing your cones, and so eventually you lose shades of some color.
yeah, any one in particular than others?
I don't know off the top of my head. But we're going to do this experiment.
So
It's green.
people that are colorblind because they don't have the center vision,
Yeah.
do they have a problem with?
I don't
Do
like
they need to
that.
wear corrective lenses then because they can't?
You can see fine, you just can't see in color. Everything's a black and white movie if you're colorblind.
But if you have nothing in the center, where they're
Well, yeah, if the cones are dead, then you'd have to use your periphery to be less acute. But if the cones just aren't working,
oh,
then you can see color. So do what this is saying. Stare at that little cross. Stare at it. And then tell me what you notice.
Green and
The
mini
circle moves when your
circles.
eyes move. The green like background, not really there,
What?
circle
And
of
it
dots.
disappears. Does it
gone.
disappear or is it just me?
Yeah, it's gone.
When you blink.
Keep staring, you notice the one, the circle.
purple
there?
circle.
It's gone.
Whoa. Whoa.
Do they do certain types of tests to see how people interpret different?
Well, the problem is, of course, that's your brain interpreting, right? So
Yeah.
You may just messed up. I don't know. But optometrists can do things with the math and determine what's going on. But the goal of this is not to get bogged down in the color. The question is, why did the purple thing become green?
Because that's what you're interpreting. Your
So you're
brain.
interpreting purple just a second
Now
ago.
it's
Why did it all of a
just,
sudden
now it's
magically
just
become green?
invisible. It's not
Because
green.
that's what color you really see.
Let's go through the word at the top of the right-hand side of the screen.
Adaptation.
We're going to learn about adaptation briefly. Important concept. You did this in lab last week. You just didn't remember that.
What?
Or on Monday, sorry. Monday. Last week, Monday.
The other day.
Last class. Okay, adaptation is when receptors fatigue. What does fatigue mean in English? They
Strain.
get tired, they get worn out like me. So, what's happening in adaptation is the receptors, not the brain, that's habituation. This is receptors just get tired of working. Right? So think about this animation. Every spot is purple, right? And you're staring at the purple as hard as you can. What do the photoreceptors do if you keep using them over and over and over without blinking your eye?
They strain.
They start to get tired. And what do receptors do when they get tired? They turn off. So your eyeball is, quote, turning off the purple receptors. Remember, there's not purple, right? It's some combination of red and green or whatever.
So it defaults to whatever, it defaults to one of the three.
Right.
That you
So the
actually
trick is,
see.
the ones that turned off, turn off, but the ones you didn't use, stay on. So, if I'm using, let's go back to the math here. Purple is a combination of kind of bluish over here, a little bit of reddish. So the only one that's left that's not getting tired is the green. So the spot shifts to green because it's the only one your eye still has on. So it sees a circle, only the green turns on, so the brain says, oh, the spot must be green, because the purple one's not firing,
Even though there's not really even a color there at all.
even though there's not a color there.
So then where do you only see that it's not a color there after you've been staring at it for a while?
Because normally your eye would never focus on a spot that long. Your eye would be shifting around, so none of the photoreceptors get tired. If you ever watch someone's eyes, especially if they're reading and notice your eyes jerk around and move around, that's the brain's way of forcing the receptors not to get fatigued. You're constantly using different parts of your eyeball. So it gives every receptor time to recharge, so you never get this effect. When you're staring here, I'm forcing you to look here with your eyes open. The receptors don't move, so eventually they get tired of telling your brain the same information over and over.
So is it the receptors that are in control of this, or your brain that's turning it on and off?
The receptor's getting tired, but the brain normally would keep your receptors moving enough to not tire. But the brain is forcing your eye to stay still.
So why does it go and then come back?
Because when you blink, then you reload the receptors and they
When I'm not
recover
blinking,
fast.
it'll do it.
Yeah, remember cones are quick. Once you blink, they reset and they go again.
Cones are quick. The glimmer in the eye.
Brain
Yeah,
versus
you've ever
brain.
seen someone with a star in their eye or something? It's just the prettiest little glimmer. What is that?
Reflection of the light.
Eyes sparkle.
I do not know that. I'll go with love.
Oh my gosh.
I do not know.
Love struck.
So here's the same thing. Here's the line that's a little more obvious. Stare at only one fruit. Start the apple with a line.
What?
Stare at it.
Whoa.
Whoa. What did the green lines become?
Green?
Red.
Oh, red.
And the apple becomes.
Green.
It's still red.
Oh yeah, what's this name?
Right, you gotta stare at it, and it determines in the person. It's a big screen.
Oh, yeah, it turned green.
Right. The color didn't actually change on the animation. What's happening is as you stare at it, you're forcing those neurons to fatigue more. The brain loses that 1/3 of the color, so the brain reinterprets it as the different color because you turned off one photocell.
So your brain is like filling in the missing.
Yes. So the brain
Information.
says, well, if I'm only getting a red receptor, it must be red.
Oh.
That's what the brain knows. So again, the brain is interpreting the information you handed.
Why doesn't it do it with the bananas? Is it just that non-existent yellow?
Because
Yellow.
you have yellow.
It will kind of be yellow already is a mixture, so usually you have enough of the others to keep it going. But you can
Okay.
play this game all day long. You can stare at something one color, look away, you'll realize you'll get an after image of a different color, or the wrong color. Right? It's this idea of adaptation. Your eye is getting tired, and so the brain is misinterpreting what the eye is sending it, because it's losing information. It's like getting half of an email, right? You can't quite interpret it, right? Make sense? So that's That's your cones. Is you're sending 3 colors to the brain. You do the same thing here. Start at the X. Right? It works better on a little screen. And notice there's an after image.
Mm-hmm.
And notice it's the wrong color.
Goes to
Whoa.
white.
Oh, yeah.
That's the same thing. You're staring at a bright image. It burns the retina. Remember when your monitor used to have that screensaver because it burned in? The concept here. You're burning in the retina gets fatigued, turns off, the brain has to interpret the other photoreceptors being the correct ones, therefore it turns on a different color.
So adaptation, is that, I'm not quite understanding. So is it the brain misinterpreting,
No,
or is it
the
interpreting?
eye turns off.
The eye
The brain's
turns off.
interpreting correctly what the eye tells it. The eye just quit telling it. So the eye is no longer sending the impulse to the brain. The brain is always correct. Right? And that's all this. Make sense? So you can do, you know, the red leaves the green leaves the red, so it's the opposites kind of thing. Make sense?
So it's the brain making up the difference?
Yeah, the brain is interpreting the difference. So if you lose the red, it must be green. And you can get all sorts of biology with why that's the most bright thing you can see, blah, 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 blah. Advertisers know this trick.
Well, how is it that we all interpret, I mean, not all of us, but the majority can interpret the same colors? So
Are you
like
sure?
we can interpret yellow, all
Are you
of
sure?
us, well, most of us. 'Cause
But
most
what you
of us
think
said
is green
yellow.
and what I think is green might
Right.
not be the same thing.
The problem is, yeah, your brain, that's the question of the land, right? Is everyone's brain just taught that that's yellow?
Right.
If you're in a jungle and I hand it to you, would you still know it's yellow? Or would your brain think it's something else? So is color acquired, or is color genetically understood? If you're colorblind,
As
there
long
is
as
no
we all
yellow, right?
agree.
I think
The
it's
same.
taught because we teach kids their colors.
That's true, if we taught them the orange was purple, then they
If they're
Would they
always
see
told
it, yeah,
wrong
that's the question,
thing,
would it look
they call
different
it
to your
purple.
eye, or would it just be different?
right.
So what about animals?
Well, animals have big, so some animals only have 2 cones, so hence like dogs and stuff can see reds and blacks and whites. But they can't see, birds of course can see everything we can see. They have all the same color. Some fish have 3 cones, a lot of animals have only like 2 cones. So
What
they can
about
see black.
bulls and red?
You asked about cows, right? Cows are like dogs, they can't see red per se, they can see the shading of red. So they can see black and white and overtones, but they can't see blue, I think it is. Don't quote me. I haven't looked at my cow, I don't know.
So don't buy your
Is
dog
that an
a
urban
white
legend
shirt.
type thing? Do they really get aggressive with red?
They get aggressive because people stab them. So
Yeah.
that's why they get aggressive.
Kind of a learned behavior then?
They're like, I'm
Okay.
pissed off.
So let's go back to here for a second. So we found our, oh, I went too far, sorry. Go this up here a little bit. So we did our codes, but then why is reading in low light difficult? So let's walk through it. So what type of things are you using in low light? Rods. Tell me about rods. They're low acuity, right? So partly you're using rods which are lower acuity. They're on the outside of the retina, right? So as you move away from the center of your view, you're doing more and more cones, more and more rods, less and less acuity, so it's harder and harder to read, right? So hence looking at stars sideways kind of thing. So periphery has pure acuity. Right, so here's the idea of convergence. The cones in the center of my eye have small convergence, meaning I get a very sharp signal. Every cone will send a separate image to my brain. Think of the pixels on your computer monitor, right? This is the old 386 I have at home kind of thing, right? It takes 50 things to make one signal, so big blocky pixels. That's the difference between the acuity and the convergence. They're opposites of each other. The more you converge, the less acute you become. Right? So you have the color concept, you have then the acuity concept, all adding up. So the center of your vision, in the center, has the highest acuity, the lowest convergence, and the most color. Right? Make sense? So that's why your brain tells your eye to keep moving as you're reading. It's trying to move your center over the images to get the full picture. Right? And that creates a problem, though. Let me show you this problem, which is this. This is actually what your eye sees. In daylight, if you're reading the text, this is all you see. You see color in the center, and the periphery is junk. Right? So why don't you see this when you're reading your textbook?
Because your brain is interpreting.
Brain's interpreting, and what's your brain telling your eyes to do so you can read the whole sentence?
To focus.
Move. So your brain takes your fovea, the center, and keeps moving it all over the image and remembering what the picture was to make the image in your brain what you see. Remember, the eyes are a camera. So your brain's moving your eyes all over the place to try to get the focus here. At nighttime, that doesn't work. So at nighttime, your brain basically is just using the periphery with bad resolution. Right? So again, the brain is trying to interpret the eye by forcing your eye to move around to get the pictures it needs to make the image. Right? It's like taking your cell phone and doing this kind of thing. Right? That's what your brain's doing with your eyeballs. The brain's telling the eyeballs to move to get the pictures on every photoreceptor it needs. Cool, huh?
Mm-hmm.
So let's do that. Let me show you a little picture here. So this is what's happening, is the brain is telling the eye to move, to focus the image on the center with the best vision. That way it gets a good signal. Right? So if you read this little blurb, just trivia. The center of your eye is only 2%, 2 degrees, 1% of what you see. It takes up 50% of the optic nerve. So half your vision is in that little tiny speck that your brain keeps moving around to find the images. Cool, huh? I think it's fascinating, but yeah.
So if it's low light, your brain's still trying to, it doesn't know, I mean, it's still moving around trying to find center even though the center's no good?
Right.
Okay.
And that's why you'll find at nighttime, if you look right at something and it's dark, you get a lousy picture. You have to force yourself to look away from the center. Otherwise, you'll get a lousy picture. So your brain doesn't know that. Brain knows cones are what matter. So you have to teach yourself to look away from something. If you take an astronomy class, I always say look away from the star, because you'll lose your vision once you look at it. Don't look at the moon, look next to the moon.
Really?
That's the concept here. But that's something your brain has to be taught, basically. Make sense? Right, and that's your receptive field and all this kind of stuff. So what we're going to do now is, now that we figured out how my cones are picking up this message in the center, they have all this nice acuity. Now we have to figure out how the message actually gets to the brain. How do I go from my photoreceptors and get a signal into a brain? So I'm going to show you this picture here. And this is going to bother you even more than the last couple pictures. Read what he's saying here. And go back to 231 and think about what he just said. Light hyperpolarizes, dark depolarizes. So go back to 231. What did it mean to depolarize a neuron?
You get the action potential from
An action potential, which means it got on, it turned the signal, right? So, look what he just said. When do your eye neurons turn on?
dark.
In the dark. When do they turn off?
Light.
This should really bother you now. When I turn on the lights, your eyes actually turn off the neurons. When I turn off the lights, they turn on. When you close your eyes, your eye comes on. You open your eyes, it comes off. So not only is the brain inventing the colors, but really you're not even there. Right? You are absent when your eye sees you. It sees the dark, not the light.
Wait,
So what are we
Oh.
one more
Okay.
time.
Yeah, you're going to stand nice and something.
I can't
So what are
process
we
your
seeing?
I know you
words
can.
that fast.
Yeah. So when the light goes down, when the light gets darker, the neurons activate. When the light turns on, the neurons rest.
Is that because they have to work harder when it's
That's just the way it is.
So what are we
Is it
seeing when it's
because
light
there's more
out?
vibration
No,
coming into
it
your
just is the
eyeball
way it
when
is.
there's light and there's less vibration?
That's the way you're built.
I don't understand.
But you know this from dark age of
Together?
the camera. When you used to have a camera, you sent the film into the lab, and they gave you two things back. What are the two things they gave you?
The negatives and the photo.
The negative, so that your eye is the same as the camera with a negative. You see the opposite of what you actually saw. You see the dark turns on the brain. The light turns off the brain, exactly like black and white film. So not only are you inventing a color, you're actually inventing what's there anyway. But we're going to go through the steps it takes to go from stimulus to brain. So there's 3 steps you're supposed to know in this process before you go neurotic and drink yourself into oblivion. Right?
How did you know?
That's why it's best just not even to learn this stuff, right? Just ignore it. Magic, witches, humors. There you go, you're done. All right. So we're going to start with light hits a receptor, a photoreceptor. This could be a rod or a cone, it doesn't matter. But I'm seeing something with my eyes on. What does light do to my neuron?
Turns them on.
Turns it off. So the receptor